Hello, my name is Bernie Eigel. I'm a medical oncologist and the Provincial Director of Clinical Research at BC Cancer. It's my pleasure to welcome you to part 2 of the Innovations in Clinical Trials Forum. We have phenomenal speakers today, and it's my honor to introduce them now. Dr. Gleave is a distinguished professor and chairman of the Department of Urologic Sciences at UBC and a British Columbia Leadership Chair. He is co-founder and director of the Vancouver Prostate Centre, a UBC and National Centre of Excellence. Dr. Gleave is a clinician scientist and a urologic surgeon whose clinical practice focuses on urologic oncology in a multidisciplinary environment spanning localized and advanced cancers. His research characterizes molecular mechanisms mediating treatment resistance in cancer, focusing on adaptive survival responses that drive acquired treatment resistance and designing combination co-targeting strategies to create conditional lethality and improve cancer control. He has patented several anti-cancer drugs and founded Oncogenics Pharmaceuticals, and recently co-founded Triad and Sustain Therapeutics. Dr. Gleave is the recipient of numerous awards and accolades that will take too long to mention today. In 2017, Dr. Gleave was appointed to the Order of Canada for his leadership role in developing new treatments for prostate cancer. I'm also proud to say that he has been a key mentor since my early career, and so it's a special privilege for me to be introducing him today. Dr. Andrew Shee completed his Bachelor of Science and medical degree at the University of Saskatchewan, internal medicine residency at Western University, hematology fellowship at McMaster University, and the transfusion medicine residency at McMaster University. He also has completed thesis-based masters in the Health Research Methodology program, also at McMaster, through the Ministry of Health Clinical Investigator Program. Currently, he works as the Transfusion Medicine Director at Vancouver Acute and the Regional Medical Leader at Vancouver Coastal Health Authority. Dr. Kevin Hay is a clinician scientist at the Terry Fox Laboratory and Leukemia Bone Marrow Transplant Program of BC in Vancouver. Dr. Hay received a Master of Science in Immunology and his MD at the University of Manitoba. After completing residency in internal medicine and a clinical fellowship in hematology at the University of British Columbia, he pursued a postdoctoral fellowship in cellular immunotherapy at the Fred Hutchinson Cancer Research Center in Seattle, Washington. Dr. Hay's research focuses on understanding the unique toxicities associated with chimeric antigen receptor T cells, as well as the development of novel CAR T cell therapies. He is director of the BC Cancer Clinical Cellular Therapy Laboratory, where he oversees the processing of cells for hematopoietic cell transplantation. He also serves as a medical director of the Conconi Family Immunotherapy Laboratory and is a principal investigator on a phase 1-2 trial in Canada using CD19 CAR T cells for the treatment of B-cell malignancies. Daniel Renouf is a medical oncologist and the executive medical director at BC Cancer Vancouver Centre. He is an associate professor at the University of British Columbia, Department of Medicine. Daniel's research interests include developmental therapeutics, genomics, and biomarker developments within gastrointestinal cancers, with a focus on pancreatic cancer. He is the leader of the BC Cancer Phase 1 program, the BC Cancer GI Tumor Group Chair, the co-director of the Pancreas Centre BC, and is the co-chair of the Canadian Cancer Trials Group Pancreatic Cancer Disease Group. Sally Prozik is a Senior Policy Advisor for Regulatory Innovation in Health Canada's Health Products and Food Branch, supporting the modernization of Canada's drug and device regulations. Sally provides strategic advice in the implementation of Health Canada's Regulatory Innovation Plan, including implementation of the new pathway for advanced therapeutic products. Prior to joining Health Canada, Sally worked in a number of senior regulatory and quality positions in Johnson & Johnson's medical device business, as well as in a startup pharmaceutical environment. Dr. Gleave will start the session off with his presentation entitled Plasma ctDNA as Liquid Biopsies to Genomically Segment Cancers and Monitor Evolution of Treatment Response and Resistance. Thank you.
Hello, my name is Martin Gleave, and it's my pleasure to present to you my perspective and our research on the evolving role of plasma circulating tumor DNA as a liquid biopsy capable of segmenting cancer heterogeneity and helping provide a tool for monitoring treatment response and evolution in real time. These are, of course, important needs from a biomarker point of view for helping bring precision medicine, and particular precision oncology, to standard of care, promising to individualize cancer care based upon a patient's tumor-specific alterations and thereby prolong lives. But there are, of course, challenges in this, since it's predicated on detecting actionable genomic alterations and monitoring their evolution during treatment. And this is not always feasible if it requires serial tissue biopsies. Over the past 10 to 15 years in advanced prostate cancer, we've seen dramatic improvements in survival through integrating new targeted therapies along the entire spectrum of the disease. But despite significant responses, resistance and recurrence occurs, and even through second and third line responses, resistance becomes inevitable. And ultimately, under selective pressures of treatment, we see different segmented populations occur that are AR-driven or DNA repair deficient or PTEN altered. And this segmentation was identified through our work with a number of different Stand Up to Cancer Dream Team collaborations and networks, characterizing these advanced cancers on the basis of metastatic tissue biopsies, and identifying the importance of biomarkers in helping us select treatments at particular times during the natural history of a cancer, in selecting new targeted or immunotherapies, and in essentially in defining and detecting mechanisms of treatment resistance. Again, these Dream Team publications reflected sequencing of specific bone metastasis, and this can be quite challenging, not practical in moving into standards of care. There's issues with regards to spatial heterogeneity. Which one of these multiple tumors are you going to biopsy, and is that reflective of the entire tumor population? And certainly you can't deal with temporal heterogeneity occurring over time, because you're not going to be able to biopsy bone serially because of cost. And this is where the promise of liquid biopsies come in. They help to homogenize this heterogeneity, and it becomes feasible over time because it's a simple blood test. So a number of years ago, led by Alex Wyatt at the Prostate Center in partnership with Kim Chi, we were the first to define the use for circulating tumor DNA signatures to define a prostate cancer genome from plasma, identifying alterations in important genes like AR, DNA repair, and others that are characteristics of a prostate cancer genome derived from a tissue biopsy. We were the first to show, using our matched sets in our Stand Up to Cancer Dream Team, that a tissue biopsy, that is a signature from a circulating tumor DNA assay, was very similar to that from a metastatic tissue biopsy in terms of mutational profiles. And even if you look at single gene copy numbers, whether or not it's AR or BRCA, they correlated highly. But overall, we found that circulating tumor DNA, when present, surveyed interpatient heterogeneity better than biopsy of a single metastatic site. From there, we went on to design a randomized phase 2 study of over 200 men with metastatic prostate cancer, randomized to two different types of AR pathway inhibitors. And we found that the level of circulating tumor DNA was highly prognostic, such that if you had very low levels or absent ctDNA, you fared much better in terms of survival compared to those with higher levels of circulating tumor DNA. So again, negatively prognostic overall. And this helped to identify men who were destined to progress early in this trial, regardless of what arm they were on. But from there, we also went on to characterize individual genes, and showed from ctDNA assays that if a patient was DNA repair altered, P53 inactivated, PI3K activated, AR amplified, and so forth, time to progression was much faster than if these alterations did not exist. So again, the use of ctDNA was prognostic in identifying particular gene alterations. They may also be used in a predictive manner. So this is an example of a heat map of many of the patients in this trial. And the most common group of altered genes with progression were AR. And we identified specific point mutations that could convert an antagonist to an agonist and help guide shifting of one AR pathway inhibitor to another. In addition, we identified AR amplification as a common copy number alteration. But in addition to the AR, many other oncogenic alterations could be detected that were important in guiding a precision oncology framework. For example, 4% had MSH altered tumors, identifying these patients as candidates for PD-L1 immunotherapy. Another approximately 20% had alterations in DDR, DNA repair, DNA damage response, and these have now been identified as candidates for PARP inhibitors. And indeed, we're working with AstraZeneca now to CLIA certify an assay that will be used across Canada to identify patients who are candidates for PARP inhibitors for olaparib and other similar PARP inhibitors in patients who are DNA damage response altered. More recently, PI3K inhibitors are being developed in advanced prostate cancer, and we're able to detect PTEN altered or AKT activated cancers that are candidates for this particular therapy. So essentially, over time, as we treat with AR pathway inhibitors or chemotherapy, regression, dormancy, progression, and resistance is inevitable, leading to outgrowths of different segmented cancers driven by different particular genomic alterations. And these can be determined using ctDNA and used to guide particular targeted therapies. We have to recognize key limitations, though. We're still only looking at DNA, and we know that DNA insufficiently predicts biology. ctDNA assays are now being evolved to incorporate epigenetic alterations that may provide yet another layer of nuance in predicting biology. But we also have to look at the long tail, the mutation curve, where many of these alterations are occurring in small segments of the population and difficult to test and evaluate in clinical trials, which is why this has led to an umbrella trial across Canada, led by Kim Chi, supported by Alex Wyatt, who's doing the ctDNA assays for the liquid biopsy approaches, where men with metastatic CRPC undergo ctDNA directed sequencing, and based upon the particular alteration are then assigned based upon their biomarker to one of many and evolving groups of targeted therapies, and allows us to better evaluate the impact of a particular targeted therapy in a biomarker-enriched subpopulation. Again, a precision oncology framework. So in summary, what I hope to show was that ctDNA signatures serve as liquid biopsies that help to segment cancer heterogeneity. This is important for molecular subclassification for both prognosis and selecting optimal drug therapy. These assays enable genomic signatures that are feasible, serial for real-time monitoring of treatment-induced adaptations. These are key for precision oncology approaches moving forward, and are also important as companion diagnostics for the development of novel targeted agents. Thank you for your attention.
Good afternoon. I'd like to thank the Clinical Trials BC Organizing Committee for inviting me to speak today at this session entitled Innovations in Clinical Trials. On behalf of the CLIC investigators, I am privileged to present our innovative network for CAR T-cell research in Canada. Here are my disclosures. Now, there are many components of the immune system's response to cancer, but one of the key effector cells is the T-cell. Generally speaking, one of the main roles of the T-cell is to recognize abnormal cells in the body, such as early cancerous or virally infected cells. However, cancers have developed mechanisms to evade killing by the T cells in our body, such as MHC downregulation, as the T cell receptor complex, as you see here, requires activation after being presented with a peptide in an MHC complex. A chimeric antigen receptor, or CAR, is a synthetic gene that is composed of a targeting moiety and intracellular signaling domains from the T cell that we insert into the T cell to enable it to recognize a tumor-associated antigen without requiring a peptide MHC complex, allowing for cytotoxicity. In order to insert the CAR into the patient's own T cells, the synthetic gene encoding the CAR is first packaged into replication-incompetent lenti or retrovirus. Then autologous patient white cells are harvested via leukapheresis, and the T cells are isolated and exposed to the viral vector in the laboratory. After a period of growth in the lab, the T cells are then infused back into the patient, typically following some lymphodepleting chemotherapy that aids in the engraftment of the CAR T cells in the body. Prior to the advent of CAR T cells, patients with relapsed refractory B-cell cancers, such as B-cell acute lymphoblastic leukemia and diffuse large B-cell lymphoma, had a dismal prognosis, with a median overall survival of less than 6 months, and only about 10 to 20% of patients were still alive 2 years after treatment. CD19 CAR T cells, or CAR T cells that are directed against the antigen CD19, which is uniformly present on B-cell cancers, have demonstrated significant efficacy, with 40 to 50% of patients alive without relapse at more than 2 years out from the infusion. This has led to subsequent FDA and Health Canada approvals for the products mentioned here, as you can see on my screen, Tisacil and Axicil, respectively. So then, what is the clinical trial need? Well, the problem is that CAR T cells are not yet commercially available in BC as at the time of the development of this project, and even to this day, they are not yet available. Canadian researchers have the expertise to make these products and many good ideas to improve them for the future, but translation to the clinic often lags significantly behind our laboratory discoveries here in Canada. Therefore, we decided to bridge the gap by building a network of researchers across Canada who would be able to translate this research from the lab to the clinic. So the idea behind this was to develop an academic model for CAR T cell research and delivery, where the gene and viral vector production were centralized at core academic facilities in the country, and point-of-care T cell processing at local hospitals, similar to how we do hematopoietic stem cell transplantation. Through this mechanism, we would be able to build Canadian infrastructure and expertise in this area and create jobs and foster innovation in the field. We entitled the strategy Canadian-Led Immunotherapies for Cancer, or CLIC. We decided to start with CD19 CAR T cells as a proof of concept, given their known efficacy and toxicity and the current clinical need that is presented to us here in Canada. We went with using a point-of-care automated CAR T cell manufacturing process on this device called the CliniMax Prodigy, as you see on my screen. The rationale for doing this was that we could then have a simple transfer of the know-how of how to use this machine to produce CAR T cells to other Canadian centers. Our team is composed of individuals from Ottawa, Vancouver, and Victoria, where the synthetic gene and packaging genetic information was produced by Dr. Rob Holt's lab here in Vancouver. This was then shipped to John Bell's lab at OHRI in Ottawa, where the GMP grade lentivirus was produced. Then this lentivirus was shipped to Brad Nelson's lab in Victoria, where the CAR T cells are produced from patient cells using the CliniMax Prodigy system I described. The cells are then sent back to the clinical sites in Ottawa and Vancouver, respectively. Although the process I described seems simple, this timeline demonstrates at a high-level view the multiple components that needed to be in place for this to happen. As a team, we took on the roles of sponsor, manufacturing, and clinical site leads, which led to lengthy contract negotiations and paperwork, as you can see from the paperwork that we submitted really in 2019. Considering that this was the first attempt to develop such a network for CAR T cells in Canada, there were many unexpected hurdles to overcome along the way. However, the CLIC-01 trial finally opened at the end of 2019. The CLIC-01 trial, as you see here, is a phase 1/2 clinical trial to look at the safety and efficacy of our CD19 CAR T cells. The defined sponsor for this trial is the Ottawa Hospital Research Institute, with the manufacturer being BC Cancer currently. The clinical trial sites are Vancouver General Hospital and the Ottawa Hospital, and we are enrolling 60 patients with CD19 positive relapsed or refractory malignancies, with a goal of 20 patients for phase 1 and 40 in phase 2 of the trial. Our primary objectives are to look at toxicity, so the proportion of patients experiencing either grade 3 or 4 CRS or ICANS, which are specific toxicities associated with CAR T cell therapy that are well known, or other grade 3 or 4 toxicity or non-relapse related death within the first 28 days. We will also then, subsequently in the phase 2 portion of the study, look at efficacy defined as the proportion of participants with a complete response or partial response at 6 months after CD19 CAR T cell infusion. Now, CLIC-01 is currently up and running and enrolling patients in both Vancouver and Ottawa, and we are happy to state that we are nearing the end of phase 1 and will likely report results of this later in the year. The clinicaltrial.gov identifier is listed below on the slide here. Although there is a current need for CD19 CAR T cells, the long-term focus of our group network, however, is beyond this, as I alluded to earlier. CAR T cells and other cellular immunotherapies are rapidly being developed to treat other malignancies, and we're working with the National Research Council of Canada to identify new cancer targets for CAR T cells and bring them to the clinic. With the NRC expertise in this realm of antibody generation, we have established a Canadian CAR T cell engineering pipeline extending all the way from target identification to the clinic. The first of such a collaboration is the upcoming CLIC-02 trial, which will use a camelid-derived single domain-based CAR against CD22, another antigen present on B-cell cancers. We have received funding from Biocannerex to complete the preclinical validation work and clinical trial protocol development, and we are aiming to start this trial in April 2022. Concurrently, we are also expanding our cell therapy manufacturing network to make this process truly point of care. As such, we are partnered with the NRC and Biocannerex to transfer the cell production technology that we have developed from Victoria to Ottawa, Toronto, and Winnipeg, and hope to have these sites on board by the time of the starting of the CLIC-02 trial. All of this work is really possible because of the hard work of a large, dedicated team. This continues to be a group effort, and it is really my privilege to speak on behalf of the work these fine people have accomplished. Thank you for your time.
Hello, everybody. My name is Dr. Andrew C., and I'd like to thank Clinical Trials BC for giving me the opportunity to.
These are my disclosures. In terms of objectives, over the next 10 minutes, we're going to explore emerging treatment strategies in pancreatic cancer, review novel precision medicine initiatives, and discuss how this has led to genomic-guided clinical trial designs. Pancreatic cancer is a common and deadly disease. It's currently the 4th leading cause of cancer-related death in Canada. It's recently become the 3rd leading cause of cancer-related death in the United States, and it's projected to become the 2nd leading cause of cancer-related death in North America by 2030. One of the reasons why pancreatic cancer is such a difficult to treat cancer is that we have limited treatment options, especially in the metastatic setting. Also, historically, a lot of our treatment decision-making was based on clinical factors such as a patient's performance status, age, and comorbidities. The good news is that the field of drug development in pancreatic cancer is really exploding, and there's quite a large number of molecules that are under development and being tested, both in the preclinical and clinical settings. A challenge, though, for pancreatic cancer has been our lack of predictive biomarkers. And for many years, we've thought of pancreatic cancer as 2 main categories: pancreatic ductal adenocarcinoma, which is by far the more common type of pancreatic cancer, and a rare subset known as pancreatic neuroendocrine tumors. Fortunately, this is changing, and the first really actionable subset of pancreatic cancer is those related to BRCA1, 2, and PALB2 mutations. So you can see on the left, the BRCA mutant pancreatic cancers have a unique genomic profile, and on the right, we know that these types of tumors have a unique sensitivity to specific types of treatment. This figure on the right demonstrates a patient who is highly sensitive to platinum-based chemotherapy with a BRCA mutation. And we know now that this is an important subset of pancreatic cancer. This trial, which was published in New England Journal in 2019, also demonstrated that BRCA mutant pancreatic cancer is sensitive to PARP inhibitor therapy. This was also an important trial as it was one of the first to be done in this specific subset of pancreatic cancer. And we know that BRCA mutant pancreatic cancers represent about 5 to 8% of all pancreatic cancers. In addition to BRCA, another important subgroup of pancreatic cancer that's recently emerged is mismatch repair deficient pancreatic cancer. Work done by the Johns Hopkins group as well as others has demonstrated that mismatch repair deficient cancers of many origins are much more sensitive to immune checkpoint inhibitor therapies. And we know that this is present in a percentage of pancreatic cancers. Our group has been quite interested in identifying other unique subtypes of pancreatic cancer. And one of the ways that we've been undertaking this study is through the BC Cancer Personalized Oncogenomics clinical trial, which is really a collaboration between oncologists as well as the Genome Sciences Center. In this trial, patients are consented with advanced cancer. They undergo a biopsy, and then the tumor tissue is sequenced with whole genome and whole transcriptome analysis. And then our bioinformatics team reads through the results and presents them as part of our weekly tumor boards. One of the interesting findings of this trial has been the identification of a novel fusion, NRG1 fusion, which is present in a subset of pancreatic cancers. This fusion can be targeted with a molecular targeted therapy known as afatinib, which is typically used to treat lung cancers. In this slide on the left, we can see PET scans done in patients pre-treatment and then after 4 weeks of treatment. And the areas that are glowing represents active cancer, and we can see that there's been a significant change after 4 weeks of treatment in both patients with afatinib. Ourselves and a group out of Germany were the first to describe this, and this has really led to the development of a new important subgroup of pancreatic cancer, which is NRG1 fusion pancreatic cancer. To try to identify further subgroups of pancreatic cancer, we formed a cross-Canada collaboration that's been funded by the Terry Fox Research Institute, known as EPIC, which stands for Enhanced Pancreatic Cancer Profiling for Individualized Care. This is quite a unique initiative as we pair genomic-based clinical trials with drug-based clinical trials, and I'll try to explain that a bit further. You can see here on the bottom left the PanGen bubble, and PanGen involves, again, fresh biopsies of patients with advanced pancreatic cancer, whole genome and whole transcriptome analysis, but also prospective clinical trial data collection, which includes CT scans done every 8 weeks. This can be done in collaboration with ongoing drug-based clinical trials. And one example in the purplish bubble above is the PA7 trial, which is a CCTG-led trial testing a novel drug therapy in pancreatic cancer. And by combining these two types of studies, we can get information both in terms of efficacy of new agents, but also specifically, do these new therapeutics work only in subgroups of pancreatic cancer? And if so, this type of study helps us to identify those subtypes. What's very exciting is how rapidly the field has changed in pancreatic cancer, where as I mentioned, historically, we haven't had any really important actionable subtypes of pancreatic cancer. And just in the last several years, we've now identified quite a number of important actionable subtypes. I discussed the BRCA and mismatch repair deficient subtypes, but there are a number of others, including NRG1, which we've also discussed. So the challenge now is, as we're identifying new important actionable subtypes, how can we best design trials to test agents in these individual subtypes? And one example of this is the CAPTURE trial, which is a Canadian Cancer Trials Group related or cancer CCTG-led clinical trial. It's a phase 2 molecular guided basket trial. So patients have genomic analysis through various platforms, including the POD program, and then based on the genomic data that's identified, can be enrolled into one of the arms of the study. As mentioned, this is a basket trial, so patients are enrolled to a unique basket based on the molecular profile they have. And each cohort within the study is defined by drug, tumor type, and molecular profile. This is quite a unique study because a number of different companies have made their drugs available as part of this study. And so to give you an example, we see the nivolumab ipilimumab arm, which is an immunotherapy arm, and this is available for all patients who have high tumor mutation burdens. And we know that this is an emerging subgroup within a number of different cancers, including pancreatic cancer. So I think what's really exciting is how quickly the field is changing, and the pancreatic cancer has finally entered the era of precision medicine. We're starting to now use tailored therapeutic strategies based on individual genomic characteristics. And because of this, it's very important that ongoing and future clinical trials are designed to take into account the unique subsets of pancreatic cancer, such as the CAPTURE trial. There are many studies that are ongoing in all comers, and in these studies, having correlative studies that are quite extensive is critical, because that allows us to retrospectively assess whether there are subgroups that seem to be more sensitive to these therapeutics. But as mentioned, I really believe and I'm excited about the future of clinical trials in this setting, as they will be defined in molecularly defined subsets. Hopefully that provides a little bit of insight into where the field of pancreatic cancer is now and where it's moving into the future. Thanks very much for your attention, and enjoy the rest of your meeting. Bye-bye.
Good afternoon, everyone. My name is Sally Prodzik, and I'm a Senior Advisor in Health Canada's Regulatory Innovation Team. I'm excited to be here today to tell you about our plans to modernize Health Canada's regulations, with a focus on making our regulations more agile without lowering our high bar of safety. In terms of context, it's clear that health products are becoming increasingly complex and more personalized. I don't imagine that's a surprise to any of you in this virtual room today. We also see that there's a broader range of innovative products out there, which can also come with a wider spectrum of risk. From a Canadian perspective, there is more demand for faster access to health products, and Canadians are looking for more information. And I think when we look back over the past almost year at the COVID-19 pandemic, it's really reinforced the need for us to have agility and flexibility in place to enable Canadians access to critically important health products. Since 2017, our department has been doing a lot of work in modernizing our regulatory frameworks, and we're truly building on that foundation to launch our next set of regulatory modernization initiatives focused on the theme of regulatory agility. This is one of the central tenets that came out of the government's regulatory review of the health and biosciences sector, where we heard from hundreds of stakeholders across the country who all pointed out that need for agility, making sure our regulations are internationally aligned and that there were no bottlenecks to innovation and economic growth. And so with that in mind, we developed our own roadmap for change, which enabled us to get a significant investment from the federal government in Budget 2019, along with some new legislative powers to be able to achieve our vision for change. We're now at the point where we're translating that vision into concrete actions. We've developed a plan that spans the entire life cycle of health products. We start off with modernizing the clinical trial framework. And you'll hear much more detail on this on day 3 of the conference, where my colleague, Dr. Daniel Keene, will be speaking in one of the morning sessions. Modernizing clinical trials is really about making risk-based and flexible regulations to support the new types of trials and trial designs that are being developed, and making sure that we are being more transparent about clinical trial information. The second pillar in our regulatory innovation plan focuses on a new pathway that we're developing for advanced therapeutic products. These products are drugs or devices that are so novel, so complex, that they fundamentally challenge our current regulatory system. And for these products, we can use a new legislative power to create a regulatory sandbox to set tailored requirements. For our third and fourth pillars, we move into the core of our licensing regime, which needs to be flexible and agile enough to oversee drug and medical devices across their entire life cycle. So being able to implement new tools to do this, such as terms and conditions on product licenses, or using decisions from other foreign regulators to enable access where there is a legitimate need. Our fifth pillar is also important in terms of our mandate. We want to make sure that Canadians have better access to information about the health products that they use. Ensuring Canadians can easily find information, for example, about a drug's prescribing information on their mobile device. Underpinning these 5 pillars, we also have significant effort underway in the department to modernize our compliance and enforcement activities. This slide provides a bit more information on each of the projects underway in our regulatory innovation agenda. As I mentioned, you'll hear more about the clinical trial modernization activities later in the conference. So in the interest of time, we'll just talk a bit more about the area where I spend a good chunk of my time, which is in supporting the development of a market access pathway for advanced therapeutic products. So why do we need a new pathway for advanced therapeutic products? There's no question that health products and technologies are advancing at an amazing rate. And it's important to note that many of these innovative products will continue to fit within our drug or device frameworks in Health Canada. But there are some technology areas that we recognize just don't fit well within our regulations. And for these, we are able to create tailored requirements, being able to work with stakeholders collaboratively to develop those requirements. So when we look at areas such as point of care manufacturing of advanced cell therapies, where the traditional regulatory requirements for manufacturing a drug in a central facility may not all be appropriate. Or in the area of machine learning AI, where medical devices can iteratively improve their clinical performance by making changes to predictive algorithms using machine learning. These are some of the areas that we're looking at and others related to our new pathway. We're also developing a concierge service to help support innovators in navigating this new pathway. So I could talk for a long time about the ATP pathway, but I will spend the last few minutes of my presentation talking about the impact of COVID-19 on our regulatory innovation plans and our next steps for moving forward. When we think back of what's happened over the last 9 months, it has definitely caused us to pause on our planning and policy development as we redirected significant resources to our pandemic response. However, in a way, the pandemic has required its own agile regulatory response and has given us an opportunity to test out some of the agile measures and ideas that we have been developing as part of our longer-term innovation plan. For example, we implemented emergency interim orders where we will be able to expand access to clinical trials as well as accelerate the review of medical devices key to the pandemic response, such as personal protective equipment and ventilators. We were able to test out some of the agilities that we plan to implement more broadly, like using terms and conditions or leveraging the use of foreign decisions. We've also piloted concepts in our clinical trial plans, like expanding the scope of investigators that can conduct trials. Beyond the regulatory sphere, there was also considerable innovation in the approaches we were taking with our guidance, with our communications, and in our engagement with stakeholders and international partners. There are lots of examples of these, but just to give you a few, in our medical devices directorate, they created guidance in very quick order to help encourage the use of innovative manufacturing techniques, such as 3D printing to manufacture personal protective equipment. We also launched a brand new centralized website for information on COVID-19 related to health products, which we found a useful way to communicate our priority information to stakeholders, including many new industry players in the health product space. Those companies that were retooling their manufacturing equipment to make masks or face shields or ventilators or hand sanitizer. As we move forward, we are really looking at the early insights we've learned through our pandemic response measures and determining how we're going to apply them to our future innovation plans. We're studying these early reflections to figure out what worked and what didn't, and what needs fine-tuning before we can implement it long-term. We are still near the beginning of our 5-year modernization journey. As mentioned, COVID-19 has impacted the timelines of our early work, but our teams are committed to driving forward with our regulatory innovation agenda to have these regulations and new policies in place by 2024. So with that, I will pause and would like to thank Clinical Trials BC and the conference organizers for the opportunity to speak with you today. And I will turn the floor back to our moderator for the Q&A session. Thank you.
Speakers for their excellent presentations. They really highlight the incredible strength and variety of research going on in our own backyards and how Health Canada is working to help increase our possibilities. Dr. Bernard unfortunately had a last-minute change such that he could not present today, and so we have a bit more time for discussion and questions. Maybe we'll wrap up a little bit early. Please submit your questions and vote on them in the live Q&A area, and maybe I'll start off with some questions of my own. Dr. Gleave, there are commercial assays available like Guardant or Foundation Medicine for genomic characterization of tumors and cell-free DNA. What are some of their limitations versus this approach, and are they complementary?
In particular in the US, where these are FDA approved and covered by insurance in the US, they've been used by clinicians with varying degrees of success. I mean, they're approved to other cancers as well, with the importance of detecting, for example, EGFR mutations in lung cancer. But in general, ctDNA has better accuracy in detecting mutations, point mutations, than say copy number or other rearrangements against the sort of signal to noise ratio problem of the background normal cell-free DNA that is usually seen in plasma as an example. A number of reports in the prostate world has raised concern about the use of the current commercial assays, for example, Guardant 360 or Plasma Success or Foundation Medicine as examples. And we actually reported in JCO, Precision Oncology, a recent analysis where we compared 24 of our samples to Guardant 360 and found in general pretty good concordance for common alterations. For example, the most common alterations in CRPC is AR copy number increases and certain mutation calls. But again, that's when the fractions are present at more than 1%. When you get into lower allele fraction mutations, that's where issues can arise. They can represent subclonal passenger mutations or false positives as examples. In addition, they can miss alterations in some common genes, for example, complex rearrangements of p53 or biallelic alterations in p10 as example, or even DNA repair defects that are clinically relevant. And so again, it speaks to the importance of tailoring a particular signature to that particular disease, because not all alterations are as common in some cancers versus others. And some of the alterations may be mutations or versus copy number alterations. And the detection of a mutation can be done at a lower fraction, percent fraction, compared to copy number alterations. So the bottom line is you have to be careful when applying a commercial assay because there are limitations.
Mm-hmm. Okay, thank you. Dr. Hay, what are the limitations of applying CAR-T to solid malignancies, and how are those being overcome?
So there's a number of different limitations to solid malignancies. One of them being, for example, the tumor microenvironment itself, which is very different and very immunosuppressive in solid tumors. And so that is a challenge that a lot of investigators are looking into, is to how to alter the tumor microenvironment to make it a more pro-inflammatory environment as opposed to a tumor supporting environment. So that's one key aspect that a lot of people are engineering CARs around. So one specific example of that is to make, instead of cars, you make trucks that are basically cars that carry a payload with them, such as a cytokine that gets released into the tumor microenvironment milieu that is potentially, pro-inflammatory and activating, the macrophages and surrounding immune effector cells there to become more, tumor killing as opposed to immunosuppressive, as we know often happens. A lot of these tumors have, high Treg populations or very suppressive macrophages. Another key problem is finding the right target that doesn't cause toxicity to your normal tissue, because CARs are kind of a little bit indiscriminate at this stage in terms of they see an antigen and then they kill everything that expresses that antigen. So the success with CD19 B cells, CD19 CAR T cells, is that CD19 is only found on B cells. So, you do have, as a side effect of CD19 CAR T cell therapy, B cell aplasia. But we know that patients can live quite a long time without any B cells, without any significant issues, and if their antibody levels are low because of their ongoing B cell aplasia, intravenous immunoglobulin can be used as well. Although whether that is actually absolutely necessary, that's a little bit of a debate in the field currently. So I think there's a few different challenges. Finding the right target and trying to find the way to get into some of these very immunosuppressive environments are two of some of the major challenges.
Thank you. Alison North has a follow-up question for Dr. Hay as well. What was the most challenging aspect of building the CAR-T engineering pathway, and what advice would you give to others attempting to build similar collaborations across Canada?
So I think a lot of the initial challenges that we experience is the idea that people haven't done this before, and so the very concept that when you approach the different health authorities, for example, saying that you're going to actually manufacture a product, they aren't used to being that sort of aspect of saying, okay, we're taking on the responsibility of a manufacturer, not just the responsibility of a clinical trial administration site, which is usually the way things are done in terms of clinical trials in Canada. So I think that's one particular thing. The other thing about building a kind of trans-Canada pipeline is working with a whole bunch of different institutions and trying to make sure all those interinstitutional agreements are in place so that everybody gets appropriately the resources they need, to do this, but also the quirks of each individual institution. So to be honest, a lot of the challenges, mainly more paperwork, mainly more legal related than necessarily scientific. But that's, one of the things that we did notice that probably led to, a year more of delay in getting off the ground than we would have otherwise if there weren't, agreements, legal agreements, et cetera, that needed to be sorted out appropriately. But now that we have that foundation in place, we're hopeful that future trials in this area that, we'll be able to lead, but also other people in Canada might do. They have that foundation to go back to help them with their development.
Okay, thank you. Martin, I think you have a question as well for Dr. Hay. You're on mute.
Sorry.
You're good.
Kevin, we've approached the CAR-T group here in Vancouver because we've been approached by international collaborators and biotech to try to bring CAR-T therapies into the GU space. And from a solid malignancy point of view, from a prostate point of view, PSMA represents a great targeted antigen to help bring payloads to tumor tissues. What's interesting is just the amount of infrastructure required to make this happen, and a capacity problem going forward. And when you look at targeted therapies in this space over the next 5 to 10 years, not just prostate but many other solid tumors are going to have opportunities for CAR-T, for evaluating even CAR-T regimens in the clinical trial space. But right now we don't have the capacity to actually even participate in that opportunity. So where do you see, how do you see that current sort of ceiling being dealt with within your group?
Yeah, so that's a very concerning thing when you see the numbers of various different CAR T-cell trials that are out there and various different indications that are rising. And you gave the example of prostate cancer, and again, that's a very good example of one where there is a good target antigen potentially there, right, as you mentioned, and so it makes logical sense to go there as one of the next kind of cancer therapies that is approaching. Traditionally, we have been administering all of these type of cell therapies as part of our FACT accredited centers, which have been, the cell therapy wards. And in particular, in Vancouver, the only clinical cell therapy administration center we have is at Vancouver General Hospital. And there have been significant, resource issues that we've had just with, maintaining our standard of care practice in terms of transplant and some of the new CAR T cells that have been for hematologic malignancies, which had been our, realm of expertise. And so then that raises the next question, well, where are all these other CAR T cell therapies going to fit? How is the program going to expand for the future? I think to some degree, this is something that has to be handled maybe at some of the higher levels of BC Cancer and VGH, et cetera, administration. So I don't want to speak out of line, so to speak, on this. But in general, we will need to think and plan ahead. We need to start thinking now about what the field is going to look like, 5 to 10 years from now, because if we don't start building capacity now, I am worried, just as you are, that we're going to get even further behind in terms of our capacity issues. I know that currently our group is trying to deal with some of the capacity issues, and hopefully we'll have a bit of clarity in terms of how to deal with some of these capacity issues, at least with the hematologic malignancies, which, as I said, have been our area of expertise. But then your specific point of solid tumors, that's definitely something that I think needs to be addressed, and that BC Cancer and and other stakeholders need to really understand how best to try to expand in this realm of cellular therapies, which is rapidly expanding. The one other particular point that would be interesting to bring up is that more and more of these therapies are being trialed in a sort of outpatient type setting, or being proposed as being administered in an outpatient setting with admission to hospital only upon, acute toxicities that potentially develop, which, may be about 50% of patients who need to be admitted. So there could be some improvements by being able to shift some of this activity to outpatient. But what I mean by outpatient is still like a full day care that patients are still being seen every day, even though they're, outpatient. So we need to think of innovative ways to address this, basically.
Okay, thank you. The next question goes to Dr. Shi. Cindy Triton asks, can you expand on the implementation study for convalescent plasma, including how it may be funded and who the other key stakeholders will be beyond researchers?
Happy to. So thank you for that excellent question. Just in regards to the why we're having a study of the study, so to speak, is that just to provide some context, when Concord was first conceived, maybe around the end of March, getting clinical trials up and running takes anywhere from 6 months to a year, realistically speaking. And by the time this was conceived, we were rolling out sites within about a month. So this all happened on very short order. And I think we're being open with ourselves, as we've been having discussions within the study teams. If we could go back in time and sort of change a couple things or roll out things in different ways, we probably would like to take some of those lessons learned and maybe apply them to future trials. So that was one of the ideas kind of underpinning this. And we're happy to report that we got a grant from Canadian Blood Services to help roll this trial out. Plus, we're working with MyTax, who is able to help us match some of the funding for research assistants and grads. Just in regards to our objectives for the implementation study, we divided this into 3 sort of sub-objectives. The first one is to do qualitative process mapping. So we'll be interviewing key stakeholders and doing structured interviews and focus groups to try to figure out through kind of qualitative analyses whether or not we can find different themes that emerge from these interviews. And then we'll also process map and present this as a unified modeling language activity diagram, which is a tool that sort of is used to sort of combine all this data with different stakeholders and diverse backgrounds. Within that qualitative process mapping, the 3 main areas we plan to focus on is, one is at Canadian Blood Services, to try to figure out the role of Canadian Blood Services and blood bank logistics in the production and delivery of convalescent plasma. The second one is in the healthcare setting, and you can subdivide that to what happens in the blood bank, what happens in clinical areas, and then what happens in terms of sort of clinical trials regulations. So we have a bunch of different stakeholders lined up there. And the last one is through public health. So essentially exploring the idea of how does public health become further intertwined in terms of rolling these trials out, in terms of helping us find donors and helping us find patients. The second sub-objective of this implementation study is, I alluded to the fact that computer simulation modeling was used to sort of predict where inventory should go, since it was fairly limited, especially at the beginning of the trial, to different parts of Canada. And this was done mostly based off of modeling that was developed at McMaster University. And the discussion that occurred was that as you apply this to other centers, such as British Columbia, the geographical expanse is different in, let's just say, Ontario and British Columbia. You have different health authority structures. It sort of changes how you might distribute some of this product. So it's to help validate their model into other jurisdictions and figure out what's generalizable and maybe what needs to be a little bit more institution specific. The last part is, when we take all these first 2 sub-objectives together, we'll put it all together in a bit of a knowledge translation document, and we're hoping to incorporate that into the Vancouver Coastal Health pandemic planning documents, some documents over at Research Institute for sort of dormant protocols, and then finally a finalized report with the stakeholder mapping that will go to different scientists of the transfusion world, as well as Canadian Blood Services. So that's a bit of a broad overview of that project.
Wow. Okay, thank you for that. Another question for you from Delia Cooper. What is the difference between convalescent plasma and the IgG infusions which primary immune deficient patients receive? And maybe I'll add on to that one of my questions in terms of how does this differentiate from an antibody cocktail like the Regeneron, and what are the pros and cons of these?
Yeah, great questions. So maybe first I'll address convalescent plasma versus IVIG. IVIG is collected from source plasma from a variety of different donors collected all around the world and usually processed or what we call fractionated in different centers in the United States and Europe. Currently, for example, in Canada, we send all our source plasma that eventually gets made into IVIG to other countries. And by and large, we're not considered, we're not self-sufficient in terms of providing our own IVIG in Canada. But I know there's some processes happening at Canadian Blood Services that are going to change that. So because of that, you may get some antibodies related or that have activity against COVID-19 if some of those donors have had COVID-19 and recovered. But by and large, as you can imagine, because it's pooled amongst a very large amount of donors, any of that antibody is going to get diluted out. So certainly convalescent plasma has a bit of a greater potential in terms of having a higher concentration of those binding antibodies and also the neutralizing antibodies to the virus. So I'm a bit biased here, but my preference is that probably convalescent plasma is probably the better therapy compared to IVIG, though I do know some jurisdictions are using or are considering running trials using IVIG in COVID-19 related illnesses. Now, in regards to where convalescent plasma measures against the antibody cocktails out there, it's unknown at this point. So certainly the advantages of having an antibody cocktail that's recombinant is that you're not reliant on donors to provide the supply of your therapy. Certainly though, convalescent plasma, since you're getting it from donors who are donating altruistically, that therapy would be cheaper. In terms of efficacy, totally up in the air right now. Those studies haven't been released, but I certainly know there are studies being planned and even considered here in British Columbia comparing some of those antibody cocktails to convalescent plasma.
Thank you. Dr. Renouf, so some of the speakers in the previous sessions were speaking to trials in rare populations with molecular subgrouping. It feels like oncology is heading in that direction too. How are clinical trials going to adapt to studying smaller subpopulations defined by these genomic drivers?
Yeah, that's a great question. It's definitely an ongoing challenge. I think one of the ways that we're looking to address this is through basket trial. The CAPTURE trial that I described in my talk, which is a national basket trial, really modeled after a trial called the TAPER trial, which is in the US, and the DRUP trial, which is in the Netherlands. And we actually have data sharing agreements with those other two trials. So it's really an international study assessing molecular subgroups of different cancers that may be sensitive to different drivers. So, I think as the field moves forward, there's kind of two main strategies. One is these type of international collaborations to assess sensitivities in different subgroups, and the second strategy is trials focused on one subgroup that might be quite rare, that has really international collaboration. And so we can think of the trials looking at NTRK inhibitors, where NTRK fusions are a rare but quite important driver in quite a number of cancer types. And the development of NTRK fusions was done in clinical trials where they received referrals from all over the world for patients to receive drug. And so I think those are two of the strategies to develop new agents in small molecular subgroups.
Okay, thank you. And sorry, there's a question from the audience. I'll try to paraphrase it. I think the question was around, it was from Tanya Bubella. I did not get a close look at the industry partners and their oncology drugs made available, but are they all already approved agents, or are you considering expanding to investigational agents as well in these baskets?
Yeah, I think that question probably is referring to the CAPTURE trial, and CAPTURE is pan-cancer, and the design of the CAPTURE trial is only drugs currently that are already approved, but it being used in off-label indications. Definitely, there's a strong interest to further develop that trial platform, as it's been quite successful, and move into a more earlier stage agents that are still in the development setting. And so to make it kind of a basket coordinated phase 1, phase 1B type trial is one of the plans moving forward. And I know there are a number of such kind of earlier phase basket trials underway. I think specifically to pancreatic cancer, a challenge in testing new combinations is that we know that the bulk of pancreatic cancers are KRAS mutant, and in the KRAS mutant setting, most targeted therapy and immune therapy combinations are not very effective. And so there's really a need still for newer drug development to target that subpopulation of pancreatic cancer. In the KRAS wild type setting, which I think is really an exciting area that has been understudied, we are finding more and more important and actionable drivers. And so specifically testing novel combinations in KRAS wild type pancreatic cancer, I think is a very exciting area within the field that's moving forward now. Just to add one, back to what Dr. Gleave was talking about the different platforms for analyzing aberrations, and I think I agree with a lot of what he was saying. And one of the challenges I think with current platforms is that they're designed in a pan-cancer way, but we know the ability to detect ctDNA that is of clinical relevance, for instance, is quite different in prostate cancer than it is in pancreatic cancer. And despite pancreatic cancer being a tumor that often is widely metastatic, we would think that it would be one of the tumors where we would have very high sensitivity ability to detect ctDNA. That hasn't been the case. And also the types of aberrations we're interested in the pancreas world are different from prostate, different from lung. And I think we need to move away from a kind of pan-cancer molecular strategy to a more tailored strategy on a specific cancer by cancer level. And I think that's some of the work that they've led the way in the prostate cancer world, and that's where we're trying to move in pancreatic cancer as well, as there's unique aberrations that we would be interested in that are different from other cancer types.
Yeah, that's a very good point. Sally, you talked about a new pathway for advanced therapeutic products. Can you clarify the scope of products for this new pathway and some examples, please?
Sure, happy to. So the ATP pathways really came out of a lot of discussions that we've had with stakeholders over the past few years, recognizing both things that are near our doorstep or at our doorstep for authorization, and those things that we don't even know exist, what might come up 5 or 10 years from now that are very complex or novel areas that we can't fit within our existing regulations. And so we have this new legislative authority to be able to essentially create requirements per product. And so you can look at the existing requirements and the regulations, pull the pieces that make sense for oversight for that product type, and then develop the other parts that are relevant. And so a couple of key areas, and we've already talked today about CAR T cell therapies, for example. When we look at point of care manufacturing of these types of products, currently the Health Canada regulations require GMP certification for manufacturing. That's not something, when you look at the differences between how certification for GMP is set up for a central manufacturing facility, not necessarily applicable at a point of care site. So being able to look at potentially the relevant quality oversight requirements that are relevant for a product, and be able to put that in place where we're not constrained by the kind of the frameworks that we have in place today. We are this year looking to pilot an ATP candidate, so we're actively working internally on kind of what frameworks are going to look like, and are expecting to do significant stakeholder consultation and engagement this year. One of our key mandates with developing this new pathway is ensuring that when we are developing the requirements for a new product area, that we are doing that in collaboration with stakeholders. It is not intended that we would, Health Canada create the requirements and then push that out to stakeholders. We really are intending to have partners around the table, subject matter experts, clinicians, healthcare system partners, patient groups, to be able to ultimately devise the right level of requirements for authorization, as well as looking to ensure support to adopt that product in the healthcare system. Hopefully that helps.
It does. Thank you. Sorry, I couldn't hit my microphone button there. Another question for you. The curse that COVID has of course created some opportunities. In my world, it's helped us start bringing some of our clinical trials activities into the current century in terms of remote monitoring and remote management of patient care and so forth. You mentioned that COVID-19 caused a pause in your work on the regulatory innovation agenda, but that it provided opportunity to test out some of your regulatory agility measures. What do you see as the greatest opportunity this has afforded your work?
Yeah, it is hard to pick one when we look at so from a clinical trials perspective, we have had opportunity. We had a interim order signed by the Minister of Health back in May of 2020, where we were able to put in pieces that we had already started developing as part of our regulatory innovation and modernization of clinical trials, things like the ability to suspend or cancel part of a trial versus all or none, being able to broaden who can sponsor medical device trials, as well as who can conduct drug trials as qualified investigators. But I think when I kind of look overall at the opportunities that it has afforded us, it is kind of our recognition that redoubling down on stakeholder engagement and the partnerships that we have formed through COVID, both federal partnerships that we have gotten closer to our colleagues in various different federal departments, as well as that recognition of working directly with stakeholders in a collaborative environment. There are countless examples of that through the pandemic, and we have strong recognition internally and with senior management on the fruits of that labor and ensuring that we're continuing that as part of our ongoing modernization work.
I don't know if there are any other questions from our speakers. I don't see any more questions in our live Q&A. We're a bit ahead of time, but when you take in Francois Bernard's time, I think we're actually running late. So I'll take this opportunity to thank everybody for attending and to thank the speakers very much for their excellent talks. And I think we can probably end the session a little bit early.
Thank you.
Okay, thank you.
Thanks.